स्थान ग्रहण करें। सदस्य श्री डॉक्टर सतीश यादव जी को आमंत्रित करूंगी कि वे अपना स्थान ग्रहण करें। इस पटल चर्चा के सदस्य श्री दर्शन कटारिया जी को आमंत्रित करूंगी कि वे अपना स्थान ग्रहण करें। इस पटल चर्चा के सदस्य श्री मोहम्मद हुसैन जी को आमंत्रित करूंगी कि वे अपना स्थान ग्रहण करें। मैं सम्मानीय पैनल सदस्यों से रिक्वेस्ट करूंगी कि वे अपना स्थान ग्रहण करें। अब मैं श्री अजय दासुंगी जी से अनुरोध करूंगी कि वे कॉन्क्लेव के दूसरे पटल चर्चा के अध्यक्ष श्री विनय वर्मा, फार्मा एक्सपर्ट, फॉर्मर प्रेसिडेंट कॉर्पोरेट क्वालिटी इन सिम्बायोटिक फार्मा लैब का तुलसी के पौधे से स्वागत करें। श्री अजय दासुंगी जी। धन्यवाद सर। अब मैं श्री प्रकाश जाटव जी से निवेदन करती हूँ कि इस पटल चर्चा के पहले सदस्य श्री ऋषि व्यास जी, फार्मा एक्सपर्ट, वाइस प्रेसिडेंट क्वालिटी एंड कंप्लायंस, सन फार्मा का तुलसी के पौधे से स्वागत करें। प्लीज सर। अब मैं श्री चिन्मय श्रीवास्तव जी से अनुरोध करती हूँ कि इस पटल चर्चा के दूसरे सदस्य श्री सतीश यादव जी, डायरेक्टर क्वालिटी कंट्रोल सिप्ला लिमिटेड का तुलसी के पौधे से स्वागत करें। धन्यवाद सर। अब मैं श्री ललित काकानी जी से अनुरोध करती हूँ कि इस पटल चर्चा के तीसरे सदस्य श्री दर्शन कटारिया जी, डायरेक्टर विंदास केमिकल का तुलसी के पौधे से स्वागत करें। धन्यवाद सर। अब मैं डॉक्टर राघवेंद्र दुबे जी से अनुरोध करती हूँ कि इस पटल चर्चा के चौथे सदस्य श्री मोहम्मद हुसैन जी, हेड क्वालिटी सिप्ला का तुलसी के पौधे से स्वागत करें। धन्यवाद सर। अब मैं इस दूसरे पटल चर्चा के अध्यक्ष श्री विनय वर्मा जी से अनुमति कि वे इस चर्चा की शुरुआत करें। प्लीज सर।
माननीय उपस्थित सभी पैनल मेंबर्स और जितने अतिथिगण आए हैं उनका सबका स्वागत है। तो इस परिचर्चा में जैसे कि आप सभी को मिले थे इथिलीन ग्लाइकॉल डाइथिलीन ग्लाइकॉल या DEG और नाइट्रोसोमेन इम्प्योरिटी बहुत बड़ा चैलेंज है फार्मा इंडस्ट्रीज के लिए। वो उसमें चाहे स्मॉल स्केल इंडस्ट्रीज हो, मिड स्केल इंडस्ट्रीज हो, चाहे लार्ज स्केल इंडस्ट्रीज हो, लोकल मार्केट के लिए और उसके अलावा जो भी एक्सपोर्टर्स हैं उनके लिए भी बहुत बड़ा चैलेंज है। तो इसी पटल में मैं चाहता हूँ कि जो हमारे पैनल मेंबर की चेयरिंग कर रहे हैं विनय वर्मा सर, सो थोड़ा सा गाइडेंस दें कि ये रिक्वायरमेंट्स आया क्यों और उसके किस तरह से टेस्टिंग रिक्वायरमेंट्स हैं और क्या रेगुलेशंस हैं उसके लिए।
Hello. Hello. Thank you, Ajay. First of all, congratulations to Sage University and Bios Laboratories University and industry both have collaborated well and organized this fantastic session and this conclave. So congratulations to entire management of all the collaborators, those are all organizations, those are collaborating for organizing this program. And thanks for inviting us and giving this opportunity to interact with all of you. आज का जो सब्जेक्ट है DEG, डायथिलीन ग्लाइकॉल, इथिलीन ग्लाइकॉल, नाइट्रोसोमाइन इम्प्योरिटीज और इनकी टेस्टिंग रिक्वायरमेंट और ये रिक्वायरमेंट्स क्यों आई, कैसे आई और क्या प्रॉब्लम है इन इम्प्योरिटीज से, ये हम पैनल में डिस्कस करेंगे। यहाँ सभी एक्सपर्ट्स अवेलेबल हैं। सो ये कोई अभी की समस्या नहीं है डायथिलीन ग्लाइकॉल और इथिलीन ग्लाइकॉल की। वे बैक in 1937 sulfanilamide elixir was manufactured by one of the corporate company. उसमें उन्होंने DEG बेस यूज किया था और उसकी वजह से करीब 107 पर्सन्स की डेथ हो गई थी, उसमें से मोस्टली चिल्ड्रन थे। ये दोनों मॉलिक्यूल जो है DEG and EG, these are actually poisonous substances and banned now to be used in pharma products, in drug products and other API or in its components, excipients. So and basically these compounds come in other components as a contamination, as a contaminant. Propylene glycol is used in pharma industry and glycerin is used in pharma industry, sorbitol is used. इन सभी कंपाउंड्स में, मॉलिक्यूल्स में ये DEG और EG एक कंटामिनेशन के तौर पर पाया जाता है। और USFDA ने इवन उन्होंने खुद एक्सपेरिमेंट्स करके देखे हैं और टेस्टिंग करके देखे हैं कि जो भी कंसाइनमेंट्स या लॉट्स जो आते हैं पॉलीथिलीन ग्लाइकॉल के, उसके हर लॉट के हर कंटेनर में भी उन्होंने वेरिएशन पाया है। DEG और EG कंटेंट्स का। इसलिए उन्होंने ये रिक्वायरमेंट अब डाल दी है कि आपको जो भी इस तरह के फार्मा कंपोनेंट्स हैं जो कि हाई रिस्क फार्मा कंपोनेंट्स हैं, इन्होंने कैटेगराइज किया है इसको हायर रिस्क फार्मास्यूटिकल कंपोनेंट्स, उनमें DEG और EG टेस्टिंग करना कंपल्सरी है बिफोर यूज। ये नहीं कि आप यूज कर ले और फिर फिनिश प्रोडक्ट में फॉर्मुलेटेड प्रोडक्ट में उसको चेक करें। बिफोर यूज इट शुड बी टेस्टेड एंड लिमिट्स आर आल्सो वेरी स्ट्रिंजेंट, 0.1% should not be more than that, ताकि ये जो लिमिट्स उन्होंने डिसाइड किए ये टॉक्सिकोलॉजिकल बेसिस पर है, ताकि उससे ज़्यादा टॉलरेंस ह्यूमन बीइंग में नहीं रहेगा। तो उस बेसिस पे लिमिट डिसाइड किए, इसलिए इनकी टेस्टिंग कंपल्सरी होती है। इसी तरह से हम नाइट्रोजोमाइन की यदि बात करें तो नाइट्रोजोमाइन का भी केस कोई नया केस नहीं है। नाइट्रोजोमाइन भी एक इम्प्योरिटीज है जो कि एज ए बाय प्रोडक्ट या एज ए कंटेमिनेंट फार्मा प्रोडक्ट्स में आ जाती है, या रॉ मटेरियल्स के थ्रू आ जाती है, या सॉल्वेंट्स के थ्रू आ जाती है। नाइट्रोजोमाइन की भी हिस्ट्री काफ़ी पुरानी है। 1956 में दो ब्रिटिश साइंटिस्ट ने इसको डिस्कवर किया था और उन्होंने रिपोर्ट दिया था कि ये नाइट्रोजोमाइन्स जो कंपाउंड्स हैं वो कार्सिनोजेनिक कंपाउंड्स हैं, और उन्होंने रेट्स पे टेस्टिंग किया था, वेरीफाई किया, और उसके बाद काफ़ी सारी रिसर्च हुई है। अराउंड 300+ नाइट्रोजोमाइन्स आर अवेलेबल, and out of these 300, almost 90% nitrosamines are carcinogenic compounds. तो आप समझ सकते हैं कि इस नाइट्रोजोमाइन्स को कंट्रोल करना कितना ज़रूरी है फार्मा प्रोडक्ट्स में। मेन रिसोर्स API मैन्युफैक्चरिंग की वजह से भी होता है, एक्टिव फार्मास्यूटिकल इंग्रेडिएंट विच इज़ द इनपुट फॉर ड्रग प्रोडक्ट्स। उसकी प्रोसेस में भी पॉसिबिलिटी है कि यदि कहीं भी नाइट्राइट्स कंपाउंड या नाइट्रोजेनिक कंपाउंड्स हम यूज़ कर रहे हैं और साथ में आपके प्रोसेस में कहीं एमाइंस जनरेट हो रहे हैं या एमाइंस आप यूज़ कर रहे हैं, तो इनके दोनों की कॉम्बिनेशन से नाइट्रोजोमाइन्स फॉर्म होता है। नाइट्रोजोमाइन इज बेसिकली N-double-bond-O compound, we all know। तो जो भी फेवरेबल कंडीशंस यदि नाइट्रोजेनिक एजेंट्स की यदि अवेलेबल है किसी भी रॉ मटेरियल में, सॉल्वेंट्स में, प्रोसेस में, और कहीं आप एमाइंस, सेकेंडरी एमाइंस या टर्शरी एमाइंस यदि आप यूज़ कर रहे हैं आपकी प्रोसेस में, तो नाइट्रोजोमाइन्स का फॉर्मेशन की बहुत हाई पॉसिबिलिटी हो जाती है। जैसे कि API में क्विंचिंग प्रोसेस होती है, यदि आपने एजाइड फॉर्मेशन है और उसको रिमूव करने के लिए क्विंचिंग किया जाता है, नाइट्राइट्स यूज करते हैं उसमें, वो नाइट्राइट्स रिमूव करता है एजाइड्स को, लेकिन साथ में दोनों कंबाइन होकर नाइट्रोजोमाइन्स को फॉर्म करता है, और ट्रेसेस में ये नाइट्रोजोमाइन प्रोडक्ट्स में रह जाता है। तो अभी ये रिसेंटली 2018 में ये कुछ वलसार्टन प्रोडक्ट में डिस्कवर किया एजेंसीज ने कि इन वलसार्टन या सार्टन प्रोडक्ट्स में नाइट्रोजोमाइन की इम्प्योरिटी का लेवल काफ़ी हाई था। तो आप देखिए, इन स्पाइट ऑफ हैविंग सो मच रिव्यूज, so much developed science and so many researchers, companies also do, and agencies while approving the product they also review the processes, but still this could not be identified that nitrosamine will remain and is available at a higher level in many products. Later on people came to know, agencies came to know, not only sartans but many other products are having this nitrosamines. So we can say science is not enough, right? It's continuously you have to do the research and try to find out that what could be possible which will not be safe for the patients. तो साइंस के अलावा दूसरा एक टूल है गुड मैन्युफैक्चरिंग प्रैक्टिसेस का, या आपका गुड मैन्युफैक्चरिंग प्रैक्टिसेस के थ्रू भी आप अपना कंट्रोल्स कर सकते हैं, आप क्रॉस कंटामिनेशन रोक सकते हैं, या जो भी आप इनपुट मटेरियल जो आप वेंडर्स से ले रहे हैं उन मटेरियल्स को आप वेरीफाई कर सकते हैं, टेस्ट कर सकते हैं, अपने प्रोसीजर्स में डाल सकते हैं और कंटिन्यूस इम्प्रूवमेंट के थ्रू इन चीज़ों को कंट्रोल कर सकते हैं। तो आज इसी के ऊपर हम डिस्कस करेंगे और फर्दर इसको इस सब्जेक्ट पे एनलाइटन करने के लिए मैं ऋषि से रिक्वेस्ट करूँगा कि कुछ और ग्रुप को बताएं।
Before starting, like thanks Ajay, the same university, like for running this program, and all the delegates who were here on the stage, and the participants. Thanks for joining this session. And at least like 6:30 के बाद में शायद ये सेशन पहली बार मैं करना होगा, so sorry for that. Okay, so as far as the PG and EG and attachment is concerned, वर्मा जी आपने most of the area आपने cover किया है. However, as जो वर्मा जी ने बताया, like PG and EG, let's start with that only. That the story is not a new, right? And it starts from 1937, US. Then Haiti, वहाँ पर भी करीब 70 बच्चों की डेथ हुआ था. Then 1960 to 98 और 99 के बीच में बांग्लादेश, इंडिया, एशियन कंट्रीज में भी बहुत सारे डेथ्स हुए. The thing is that it's not that ये कुछ नया है, and the problem statement is that हमको सब कुछ मालूम है, then also we are not taking care of that particular issues and incidences होते जा रहे हैं, right? Here, if you go through the incidents, what we come to know is that problem क्या हुआ था, that we cut the corners. यहाँ पर मैं पवन जी का नाम लेना चाहूँगा. मैंने आयुष का सुना, आपका काफ़ी रोचक और काफ़ी ऊर्जावान था वो, और आपकी बहुत सी बातों से मैं सहमत हूँ. And the biggest thing is what you were saying, that an entrepreneur should be able to differentiate between investment and the overheads, right? So overheads क्या होते हैं और investment क्या होता है वो हमको मालूम होना चाहिए. Companies में ऐसा होता है that जब रूटीन में सारी चीजें चलने लग जाती है तो we start cutting the corners, right? चलो reduce testing में डाल देते हैं, fine? जो vendor है उसका COA तो available है, right? सरकार मुझे COA दे रहा है, and this particular incident which has happened in India, Zambia, and then और भी 2-3 कंट्रीज में हुआ है. This starts from there on. सप्लायर ने किसी का COA लिया, उसने अपने लेटर हेड पे COA बनाया, तो कंपनी ने सील किया, उसने टेस्टिंग नहीं किया, and now all those legal and all those actions have been started. However, when someone is not doing their job properly, how it impacts to the industry, मैं उसको थोड़ा सा like सभी का ध्यान खींचूंगा. Now, regulator has come even for India के 1 अगस्त के बाद में none of the company can export any suspension or syrup or the cup syrup without testing in the Indian approved, CDSA approved laboratory. अभी सिचुएशन ये है कि आई थिंक मेथड्स भी अवेलेबल नहीं, राइट? जो लैब्स को कॉन्टेक्ट किया है इंडस्ट्री ने, जहाँ पे गवर्नमेंट ने टेस्टिंग के लिए बोला है, मेथड्स वहाँ पर भी नहीं. कुछ लैब्स बोल रही है कि मेथड्स अवेलेबल है, those methods are the pharmacopoeial methods of the raw material. Now, वो एक्सट्रैक्शन हो पाएगा नहीं हो पाएगा ड्रग प्रोडक्ट से, रिजल्ट सही होगा नहीं होगा, फेल होगा तो उसका इन्वेस्टिगेशन कैसे करोगे, आपका मेथड यूज करना है या एक का मेथड यूज करना है, वो सारा अभी कंफ्यूजन एंड द एक्सपोर्ट हैज बीन स्टॉप राइट. सो होता वही है, सपोज समवन इज नॉट फॉलोइंग देयर अकाउंटेबिलिटी, इट इज प्रिस्क्राइब्ड एज पर द रूल्स एंड रेगुलेशन एंड द इम्पैक्ट इज बिगर देन दैट. बिकॉज यहाँ पर क्या है, वो जो भी कंपनी है बट यहाँ पर नाम आ रहा है इंडिया का राइट. और हम लोग बड़ा गर्व फील करते हैं अपने आप को कहने में कि इंडिया इज द फार्मेसी ऑफ द वर्ल्ड राइट. सो आई नॉट गो कि हर कंटेनर का टेस्टिंग करने के लिए बोला है, हर कंटेनर का लिमिट टेस्ट होना चाहिए, आईडी होना चाहिए. इफ देयर आर 3 or 4 टेस्ट देन यू कैन नॉट रिड्यूस टेस्ट एज फार एज दिस पर्टिकुलर लिमिट टेस्ट इज कंसर्न. I'll keep something so that मेरे साथी के मुझसे बात कर सके. But this is what is the conflict happens when someone is not performing their own duties and then burden comes to whole industry. And then जब पूरा burden आ जाता है, it's not about कि कौन सही कर रहा कौन गलत कर रहा, then everyone need to follow that particular line of action. So PET पे I'll not speak more, and my colleagues will cover the remaining part. As far as nitrosamine is concerned, again जो वर्मानी ने बोला है, like identification was way big. However, as far as the regulation and all is concerned, 2018 से ये पूरा चालू हुआ है. नाइट्रोसामिन is way bigger अभी की डेट में compared to PEG में, right? Like impurities का start हुआ 3 impurities, then 5 impurities, then 6 impurities. अभी USFDA ने list किया about 7 impurities. And latest अभी 7 जुलाई को इसका EMA का जो लेटर आया है QA ने, they have listed about 84 nitrosamine impurities. But again, see, as far as nitrosamine is concerned, there is a huge noise, right? But यदि अभी हम देखेंगे कि हमारे घर में जो पीने का पानी आता है, उसमें भी उतना ट्रेसेस आपको मिलेगा, because it goes through the chlorination. However, as far as the responsible industry is concerned, as a pharma industry is concerned, onus remains on us, and that's the reason, as far as nitrosamine impurity is concerned, what regulation says is that पहले आप evaluate करो, evaluate in the sense that आपका जो input material है, आपका जो key starting material है, then a full chain of custody from start to end the process, right? Even DF and DF का packaging, right? So full chain of custody का पहले हमको evaluation करना है, that's the number 1. Then based on evaluation, wherever we find the risk, उस risk को हमको assessment करना है based on the testing. Based on the testing, whatever results we have, whether it is there, it is not there, from which process it is coming, from which input it is coming, input material it is coming, or final packaging material is coming, then we need to mitigate that particular risk. So that is what is the 3-step process regulators has proposed. Now we can name all the regulators, be it USFDA ho, EMA ho, and Visa ho, everyone. And so that is what is the process like all the industry is following. And many in last 3 or 4 years, बहुत से जो key suppliers है API के, and they have changed their process, they have changed their filtration mechanism, and for the entire process to see that वो avoid किया जा सके nitrosamine impurities का level. And then finally, like as far as the which countries are concerned, वहाँ तो बहुत सी companies का like export भी stop हो गया था बीच में, and now they have started along with this particular risk assessment and everything. So with that, like I will hand over to Satish or Dr. Satish, sorry.
So thank you so much, Satish. So I think first of all I wanted to thanks to Sage Group, because I think this is first kind of event is happening at Indore, where the academics and the industry, especially coming together for solving the problem. I think that's a great approach we have. I'm really clear, and the way this criticism is going on to the whole world, for the specifically for this easy issue as well as the nitrosamine. And we are Indian, so it's very important, and we are responsible because even we are working in a pharma company as a pharmacist. It's our duty, our responsibility to give the medicines to the human which are safe for their life. So that's I wanted to reiterate on this statement. Because the way this criticism is going on to the whole world, and if you read the media, social media, there are a lot of breaking news, and everybody is criticizing, specifically recent issues of the disease. They are criticized many companies from India. Why it is criticized? Because see, issues are also going to come from India because we are the leader. We are the leader who are providing medicines to the whole world. And we see how the COVID situation India needed the vaccination program. So that's very important that our name also we should protect, and that's the reason this is a very good time to introspect ourselves and see where we are lacking. And from my point of view, I feel there are three aspects we need to take care, so which I think currently we are missing. The one thing is the knowledge. I think the way we have, maybe the people who are working in the pharma, leaders and the top management, they know very well what are the rules, what are the regulations. But sometimes on the shop floor, when this time is come to implement these rules, sometimes quality get compromised. And as we seen in the herbal session, there was a lot of discussion happening how the cost cutting. Sometimes people or management thinks about the quality. Sometimes we compromise the quality, and then these issues lead into these kind of bigger issues. So I think one thing is the knowledge how we are implementing the pharma company on the shop floor. Another one is the how we are working on the quality. And third thing is how cost, how budget you are assigning to the quality. These three things I think we need to really work. And as a pharma company, we are working in a serious business. I think it's a time to balance all this and balance the quality of the medicines. So that's very, very important topic. And thanks to this bias team also to come together, collaborate with the SAGE group. And that's the reason all these experts are there in this today's event. And since both our panel members already covered the history of background as well as why this requirements come, so I'm not going to focus much on this particular point. But I wanted to reiterate on this specifically the technologies what we need to use or what are available. I think that also we need to have a knowledge, and accordingly the right technologies if we are placing into our labs, I think that will really help to control our quality of analysis. The first part I wanted to talk about mainly the easy and easy technique. So if you refer the pharmacopoeias, USP also come already with common methods. USP Journal chapter 469, which already having the common methods on the GC, specifically to analyze DG, EZ as well as triethylene glycol. These are the main three components which are coming mainly in the glycerin, as well as propylene glycol, sorbitol, mannitol, PEG, these kind of the contaminant excipients. These are getting used and having the rest of the disease. So specifically, these are the highly volatile solvents, the solvent impurities. And this chapter is not a new one. If you see ICH and 7, so many decades this chapter is there. Even as said, 1937, US was faced these difficulties, and they were having more than 100+ people got died because that time the technology was not there. But today, if you see the technology what is available in the market, you can go up to PPP level, not one level also you can achieve. And that's how we need to gear up our labs to have this kind of technologies in the lab. And you can refer general chapters, there are a lot of methods available only on the social media. But general chapter USP 469 already says about the easy common method for these kind of contaminant excipients to evaluate for the disease. It is the back column as well as the capillary column. These methods are already given which you can run on the normal CC as well. And they are also capable to go up to 100 ppm level down for these kind of methods. But I think the control strategy for these contaminants also is very important because some of the monographs still they are not defined and having limits up to 0.25% for the disease. Suppose one of the component EZ or EZ, you are releasing up to 0.25%, that may be maybe some of the cases, some formulations having the higher content of this glycerin or glycerol or mannitol, that will really create a challenge and it may go into these products into quality issues. So I think that's the reason it's time even the pharmacopoeial monographs some are relaxed. It's very important to keep these limits very stringent. And that's the reason I think we need to work on to keep the limit at maybe 0.1% or below 0.1% if it is possible. And the methods are capable, I think we should keep these limits as much as possible on lower side. Another thing is also for the nitrosamine also you cannot run on the HPLC or normal CC. And these techniques, whatever are available, I think they are very high level techniques we require to use to evaluate our products as well as the input APIs for the nitrosamine, mainly on the LC triple quad, LC MSMS I will say, or LC HRMS. These are very highly sophisticated instruments, and they also require highly sophisticated environment also. Because some of the time when you install this LC HRMS as well as LC MSMS in your normal laboratory environment, there are a lot of challenges what we see while you are validating these kind of methods. So these, when you are qualifying these instruments, really the environmental care also need to be taken care where you cannot prepare the sample solutions in the lab. And these instruments require dedicated area and which also requires the environmental care also free from contamination. So that's how these instruments installation qualification also requires to take care. Many times these common methods, whatever defined in the USP 469 or some of the general methods which are available on the media, sometimes these are not workable to our excipients and EPAs. Sometimes tuning is required, and that tuning, if suppose you are doing in the methods, these methods are required to be fully validated, and that you cannot maybe cover some short parameters. So if these methods, wherever you are fine tuning for the nitrosamine as well as these methods are required to be fully validated, else there will be a compliance issue. So that I think we need to take care. So this is what is my learning and I wanted to share to the team. But overall our whole objective in the pharmacies, we should give the right quality of the medicines to the humans. The humans are having the right to get the right quality of the medicines. Thank you very much.
Thank you, sir. Sorry for the introduction. Now I would like to request Dr. Abhay Kumar from RRCAT to join the panel discussion. So I request Dr. Abhay Kumar, who is having Design and Manufacturing Technology Division at RRCAT, for the panel discussion. Thank you, sir.
हाँ बेटा
Thank you, Dr. Satish. And good evening, everyone. My co-panelists, they have highlighted the issue very well. The issue is burning hot and important as well. And it is serious also. Because quality of medicine is no doubt a serious issue. But then we have to understand and see the things in totality. A problem which erupted in 1937 in USA, and in 2023 May, they have issued a circular which says that most of the American companies are not testing the solvent for DEG and EG, and they have made it compulsory for them to test in USA, not in India, in USA. It is a guidance which is given in May 23. So there was not an issue of this contamination in process probably, but something has gone wrong in 21 because of that the problem is there. So we have to investigate that problem, that what went wrong and how it happens. WHO intelligence is saying something different. Their intelligence is saying that some traders have contaminated or adulterated the PG or other solvents with the EG and DEG knowingly. And that particular adulterated material which has been purchased by the industry and used without testing is the cause of problem. And they are suspecting that such adulterated product is still lying in various warehouses across the world. It is not only in India, it is across the world problem. So they are now making it compulsory for EG and DEG to be tested in every solvent. Probably the manufacturing process of PG and other solvent which we are using regularly and which we are using since years is safe. But because of some greedy person, there is some problem which has now created a problem, and because of that there is a major reaction among the regulators across the world and as well as industry. So we have to see in totality and react sensibly. Coming to the nitrosamine, nitrosamine is also a well-known product. It is indeed a carcinogenic, but it is found in water it is found in vegetable, it is found in dairy product, in meat, in processed meat, in fried meat, and in many other things. And it is said that it is carcinogenic. It may be carcinogenic. They are not saying it is carcinogenic. The paper says it may be carcinogenic if taken over a prescribed limit for long term or time. So, and then it also says that if it is taken under the prescribed limit or under the acceptable limit for 70 years, it may not cause cancer. And what is this acceptable limit is not disclosed. I could not find in 2 days the acceptable limit, what they wanted to say. But it is there. So they wanted to test it, but the things are not clear. Nitrosamine, how it is produced, from where it is coming is not known. Now, agency as well as industry, they want industry to participate and come with the source of the nitrosamine, which is known till now. But they wanted industry to control it. They wanted to check industry to check it. When they are not able to disclose the limit, means acceptable limit, which they say if it is taken under the acceptable limit for 70 years, it is not giving the problem. So there are many grey areas in this also. So we have to see, means regulator has to see, lawmaker has to see this aspect as well. Thank you.
Thank you, State University. Thank you, Pakistan, for organizing this. Good evening, ladies and gentlemen. Okay. I think background of this issues, what's the regulations, the testing requirements, and things are covered. So I would like to cover what is left. So let me start with something which is there in the guidance and which is something beyond the guidance. Now, for example, if you take ethylene glycol and diethylene glycol, that's the most discussed topic today because there are at least 100 deaths now all around the world, and India is, being discussed in the wrong way for these kind of things. As my friend said, EG is coming along with various compounds. For example, glycerin, propylene glycol, sorbitol, maltitol, mannitol, XYZ. There is a big list which is given in the USP, and this is not something which came today. This USP has a list since long. There are two possibilities for these impurities to be in these risk substances, what they call in USP. One is, as Sir said, it could be an adulteration knowingly, or there could be a formation in the process itself. So, as a pharmaceutical manufacturer, we have to take care of both. So, that is why the pharmacopoeia has kept a limit of 0.1% in the raw material, and in order to detect any adulteration or mix-up, that is the reason now recently the FDA guidance has very clearly said that each and every individual container has to be sampled, and the testing also should happen on individual containers. So I think we have to underline this. This is very, very critical. For example, if we are sampling individual containers and then testing the composite, we may miss it. So the requirement here is, since it is highly dangerous, US FDA has released a guideline a couple of months before, and it has made mandatory that each and every container has to be tested individually. So this is very, very strange. First time I am hearing in my experience that each and every container has to be tested individually for EG and EG container. So when I say this, now I am going a little beyond the guideline. Probably the people who are in the development of products has to look into this. As of now, the regulations or the guidances talks only about the control in the raw material. But for example, assume that today the acceptable daily intake of, for example, EG is 0.5 milligram per kg. This is a fact, right? Now, I am controlling this in the raw material to 0.1% level. I am meeting the USP done. Now, the problem comes, for example, assume that we are using propylene glycol or glycerin in a liquid formulation. And there are formulations where this propylene glycol and glycerin are added more than 50% in the formulation. It is used as a medium, right? So in this case, can anybody tell me with the 0.1% limit is sufficient? Definitely not, right? So my request is, following USP is good, but we as a pharmaceutical manufacturers, we have scientists, we have champions in the industry, we have a good quality QC laboratory, we have the QA people, so we have to think a little beyond guideline. So just following a 0.1% limit will not be sufficient if the formulation has more amount of these risk substances. For example, I can quote you second example, right? So what is I am underlining this is very, very critical. Please, whoever is developing formulations, especially liquid formulations, please look into this. The second point what I would like to say, which is again beyond the guideline, is the USP has listed few compounds which are having a potential risk of having the residues of ethylene glycol and ethylene glycol. But there are certain things which are even beyond this. For example, one compound what I remember is polysorbate. Polysorbate is not in the list of USP, right? But the process of polysorbate can produce EG and BEG as an impurity. So again, my second request is, following USP is good, it meets the requirement, but as a responsible pharmaceutical people, we should even go one step beyond and ask the excipient manufacturer, what are the possible impurities in your process? Can EG, BEG form in that? It's very simple. A person who is in chemistry, he can very easily say that just by looking at the process of polysorbate, he can say whether it can form EG, BEG or not. So in such cases, we have to go a little beyond the guidances and regulations, and we have to test such materials also as a part of. Now, a third point again which is beyond the guideline. As I know, there are no regulations which says to test EG, DEG in the finished product. I don't think that's a pharmacopoeia which for a liquid product which says you test EG, DEG. Again, and there is no limit which can be given, and the limit is to be derived based on the formulation what we have. For example, as I said, the acceptable daily intake for EG and DEG is 0.8 milligram per kg per day. Assume that I am taking that 5 ml dose 3 times a day. For example, I am taking the liquid 5 ml dose 3 times a day. Then it is my responsibility to calculate for 15 ml what is the acceptable intake, and that should be a limit in the finished product. I am telling you, there are some regulatory agencies who has started asking questions on this, and some companies have got queries as well. So beyond raw material, if we are using this risk substances at a higher percentage in our finished products, we have to have a safety-based limit in our finished products and test that as well. Okay, so these are the 3 points I would like to tell you on the EG and DEG things. And for nitrosamine, I would just like to add one point in the interest of time. So many countries have given deadlines for confirmatory testing. So if I say nitrosamine evaluation and testing, there are 3 stages which is defined. Number 1 is risk assessment, theoretical risk assessment, if the process can form these impurities. The second is confirmatory testing, where we have to test 3 batches, minimum of 3 batches, in order to see if there is a presence of nitrosamine, right? When we say 3 batches, we have to use our own logic. At least we need to use 3 batches where 3 different APIs are used. There is no point if we test 3 batches with a single API input. That's of no use. So that is the logic we need to apply. Then thirdly, many countries have given their deadlines for filing and adding this nitrosamine into the specifications as well. As we speak today, almost all the countries, the deadlines are crossed. Most of them were completing in October 22. I think US is only allowing till October 23. I'm not sure about it. Let me check the data. But I think only US is still there. But when it comes to the limits, which is to be in the finished product, there's a clear guideline, there's a clear deadline given. But unfortunately, there is nothing being discussed on stability. So, if a nitrosamine can form in the process, there could be also a slow degradation where components in the API can convert into again secondary amines, tertiary amines and quaternary amines, and in the presence of nitrate which is in the process, there could be a slow reaction and which is theoretically very well possible. So considering this, testing nitrosamine in the stability is becoming mandatory. So we need to have a minimum of 3 batch data, and our annual stability program should include nitrosamine in the stability program if the product is prone for nitrosamine formation. The other part which I want to add is the cleaning requirements. So currently, if a product is having a risk of nitrosamine, so we are testing it manually, we are filing it because that is a requirement. I'm not sure how many people are ensuring that the nitrosamine residues are not left behind in the process equipment post cleaning. So this requirement has already come. Already there are observations around this, and also as a pharmaceutical champions, we have to ensure that we fix cleaning limits for the nitrosamine residue. If a nitrosamine risk product is made while cleaning, we will be testing the product residues. Along with that, we also need to test the nitrosamine residues. So, and we have to validate the process. So this is the requirement, and already there are observations around this, and I am sure that any regulator who is going to come for inspection is going to look for this. Thank you very much.
I need to present my slides because this topic is quite allied to this audience. I think it will be a surprise to you, so I think it's better that I use my slides to explain the topic. I prefer